So the yeast data, let me go ahead and load them because it takes a moment. Let me get them started loading, and then I'll talk about the data a little bit. Change directory. So I mentioned this a couple of times now, I think, but under data, under microarrays, there is yeast, and if you download this, you get a big zip file. Unzip it, and you get a folder that's called raw, and inside raw are all these text files. If you look at one of these text files, you have all the raw information that came out of this instrument. So you had a little chip, thousands of spots on the chips. Each spot corresponds to a gene, roughly speaking, and then the intensity for that spot tells me about the activity level or the expression level of that gene for this particular individual or sample. We had a bunch of metadata up at the top, things like who ran the instrument, what sample was on this instrument, what category of sample was on this instrument, and so on. And then we had, for each of thousands of spots on the array, information like where on the genome does this gene live. Those are the base coordinates from 393.118 up to 394.854. That's where this gene lives. Here's what this gene is involved in. Here's the chromosome that it's located on, and so on. A bunch of information. If you scroll way out to the right, eventually you find the numbers that we're going to be interested in. So let's see where they are. I talked briefly about this in the lecture that I posted for Friday. Oops, where'd it go? But you have intensities. You have an intensity for like the background level of intensity. So things that hybridize to the chip that shouldn't have hybridized to the chip. And then you have intensities for what you expected to hybridize to the chip. There's two channels on these array types. So there's a channel for control samples. There's a channel for target samples. And you can distinguish the two channels by the color of light that they emit whenever you image these. So there's channel 1 intensity and there's channel 2 intensity. Channel 1 and channel 2 background. There's mean intensity and there's median intensity. Each spot is composed of lots and lots of little probes that are specific to that gene. And so the gene copies will come and stick to one or more of those probes, and then we image them and we get a light that is a function of how many copies of that gene or the mRNA for that gene bound to that spot. So the actual number that I can pull out for this spot is actually lots of numbers which get quantified as pixels. So this is a snapshot, an actual image of these microarrays. I don't know if I've shown you this picture before, but looks like this kind of thing. Thousands of spots, and for each spot I get a light. Here's a two-color array where there's green and red, and the relative abundance of the two different colors tells me about the relative intensity of whatever went into those two channels. So if I were to zoom in on one of these spots, there's a bunch of pixels making up that spot. And the actual number that gets returned to me, like in one of these files, needs to be some summary of all those pixel intensities. And here I could either have the mean or I can have the median. We're going to work with the median. Okay, so I've read in all these files. Each one of these files corresponds to a microarray. And then here's what we said we know about these data. If you were to go through the metadata in each of these text files, you would see things about, like I said, who ran the array and what strain of yeast was on this array. So these data, they're about yeast, and what was specifically of interest was, we can find out by looking at the source for these data. These data came from a repository called Geo, and if I plug in this accession number, I get information on this experiment. So here's the abstract from the paper these were taken from. They were interested in comparing the copy number data among different categories of yeast. So fuel ethanol yeast is one category of yeast. They're used in the production of this type of fuel. There are other strains of yeast, like strains that are used in the production of wine, strains that are used for beer, baker's yeast, and I think that was it. And so they wanted to compare the copy numbers, quantifications, of fuel ethanol to these other categories. So each of the arrays, if I look at the category, will say either control or ale or wine or fuel. So I have one array for each sample, and I'll have multiple samples for each of those categories. I went through and pulled out all that information. So the file that's given by 69284 is control versus control. Remember, there's two channels, so I can put two different things on each of these arrays. Most of these have control in one of the channels, in channel 1, and then the target group in the other channel. There's two arrays that just have control versus control. This first one was that. The strain was control. Channel 2 was control. The technician was named Dunn, and this array was prepared in 2006. Here's another array that had on channel 1 strain PE2-1, which is a fuel strain. Channel 1 was control. The technician in this case was Stambuk, and this array was prepared in 2006. All the way through all of our 23 arrays. Here I'm going to collect all of that meta information. So I'm going to create a data frame that has columns for strain and technician and year of preparation. So it looks like a 23 by 3 matrix. Sample 1 was control versus control. Dunn made it in 2006, and so on. Here was an array for which I didn't know the technician or the year. Here was an oddball array for beer that was really old relative to the others. It was constructed in 1999. Everything else was much later than 1999. Here's another control array for which I don't know the technician or the year. I'm going to throw out the 1999 array and the two control arrays for which I don't know technician or year. I wouldn't really trust an array that is that much older than the others, 6 or 7 years older. Things have changed a lot over that time period, so I would expect there to be systematic differences between arrays that far apart in production. So I'm going to throw out the columns that correspond to those 3 samples. There's also an issue that each array has thousands of spots, but some of these arrays have a different number of thousands, so that some of the arrays don't have the exact same features on them. We talked about Affymetrix probe sets, for example. Each probe set has a code, and so these would be arrays that might have some differences in the probe sets that are on them. In order to simplify what we're going to do, I'm only going to focus on the spots that are the same across all 20 remaining arrays. So that's what I'm doing here. I talked about this on Friday. I'm just pulling out the spots that are common to all of the remaining arrays. There's now 6,740 spots that are common to everybody. So each one of these data frames has 6,740 spots. And I'm going to put them all together into a 6,740 by 20 matrix of intensities. The intensities are going to be median intensities. So back in that text file, we had a column for median intensities. We had a column actually for channel 1 and channel 2. And what I'm going to work with is the channel difference, actually log channel ratio. So difference in log channel intensities comparing the target channel to the control channel. Y1 is the collection of intensities, median intensities for channel 2. Y2, actually I think this might be the control channel. Y2 is the median intensities for the target channel, and I'm log transforming both of those, and then I'm taking the difference between them. Now I have a 6,000 by 700 or 6,740 by 20 matrix of intensities. Log median intensities. That's what it looks like, a bunch of numbers. Bigger numbers indicate greater, in this case, copies of a particular gene. We made these on Friday, some basic exploratory pictures, side-by-side box plots of the control channels. Every array had control in one of its channels, and they all kind of line up well. Array 13 looks a little bit different. Meta 13 was a control array prepared by Dunn in 2006. Looks a little bit different from everybody else. Don't know why. And so on. So see Friday's lecture if you want to see these differences. What we saw was there were lots of differences between strains. Here's side-by-side box plots comparing the different strains. So I averaged up the intensities of across samples of the same strain. And you can see the box plots are shifted with respect to one another. The fuel samples looked different from the control samples, which looked different from the baker samples, looked different from the wine samples. I threw out the beer sample. The beer sample was the 1999 array, so we only have these 4 categories. Interesting was that there was also systematic differences between the technicians. So here are box plots of the intensities on the arrays prepared by Dunn and Stambuk. There were different flavors of arrays prepared by these different technicians, control, fuel, and so on, but overall there's a difference between these technicians. That's a little bit worrisome. There were also substantial differences by year of preparation. 2004, 2005, 2006. Ideally, we would see differences between the biological conditions, but not between technical things like this. So, clustering. Let's look at the result of a clustering and then go talk about clustering in more detail. This is going to be what's called hierarchical clustering using Euclidean distance and something called average linkage. Let's make one plot first. The hclust function is what we use to do this. And then here's the main output of a hierarchical clustering analysis. We get this tree thing called a dendogram. And the way we interpret this is samples or features, whatever I'm clustering. In this case, I was clustering samples, so columns. Columns that are joined together low down are more similar than columns that are joined together further up. So samples 2 and 9 looks like they were joined first. They were the most similar out of every pair of samples. Same thing with 6 and 18. They were very similar. And then what happens is we join the nearest pair, and then we look at distances again after joining that initial pair, and we join the next nearest two things, which might be involving that cluster of two things, and then we redo it, redo it, redo it. Every step along the way, we're recomputing all these distances and joining whatever's nearest to one another. And then we interpret this as, here's a grouping of samples that are separated by relatively short vertical line segments. And this group is joined with that group by a relatively tall vertical line segment. So the fact that these guys are packed in pretty close, these guys are packed in pretty close, but they're separated by a fairly large vertical distance. I had to travel a fairly large vertically distance in order to get from these guys over to these guys. That would be an indication of clustering. The left collection clustered separately from the right collection. And we can go through similarly. These guys looked different from this collection in total, and these guys all looked different from these guys. So depending on where I want to draw my horizontal line, I have a collection of, quote, clusters. And if I looked into these different samples, sample 11 joined with 1 and 7 and 6 and 18, for example, could that be all the fuel samples? And here's all the wine samples, and here's all the control samples, and so on. That would be interesting to be able to verify something like that for my data. Or might it be, here's all the 2005 samples, here's 2004, 2006, and so on. Clustering allows me to pull out structure, like large-scale structure, from multivariate data. So cluster analysis is about grouping objects together. In our case, it could be grouping the samples, the columns, or it could be grouping the features, the rows. Either way. But it's a process of doing that. Generally, cluster analysis is about that. There's different approaches to doing cluster analysis, different strategies, different algorithms. We're going to look at two. One is called hierarchical clustering. That's what we just saw the result of there. Gives rise to a dendogram. There's two flavors of hierarchical clustering, agglomerative and divisive. Agglomerative is what we're going to focus on. This basically says step one in the algorithm is to allow every feature or every sample to be its own, quote, cluster. Everybody's separate, distinct initially, and then I start joining, moving up. As opposed to divisive, where I start out with everybody grouped together and I start pulling out individuals who look different from that collective. This picture was the result of agglomerative. Everybody's different. Let's build them all up into similar groups. So things that I have to think about when I do clustering, hierarchical clustering in particular, one of the main considerations when I'm doing this is how similar are, in this case, two samples to one another. 2 and 9 were similar. Were more similar to one another than 4 and 14. How did I quantify similarity? That's where I have to define a similarity or dissimilarity metric. What do I mean by similarity? And there's different ways of defining that. What we just did was the default choice of Euclidean distance. So the Euclidean distance between two vectors, it's just the square root of the sum of the square differences component-wise. So go feature by feature, gene by gene, take the difference in intensities between these two samples, square them, add them all up, take the square root. That's the Euclidean distance between samples I and j. There's different distances. There's something called the Manhattan distance, which is the sum of the absolute differences. There's the Mahalanobis distance, which is basically like a scaled Euclidean distance. There's a way to use correlation as your distance metric so that you call things similar depending on how correlated they are with one another. Just depends on what you're interested in and what you're doing. We're going to focus on Euclidean distance. That's the default choice by most methods. That's the way it is with H-clust. So that's the first thing we have to define. The second thing we have to define is called the linkage. And what this speaks to is, so if I'm comparing sample to sample, just pairs of samples, which is the first step, go do all pairwise comparisons between samples I and j. In this case, sample 2 and 9 had the smallest Euclidean distance of all pairs. But once I've joined 2 and 9 together, I now have 19 individuals, or let's see, 18 individuals, and this cluster of 2 individuals. So I can again do pairwise differences or distances between those individuals. That's the exact same calculation that we did in the first step. But how do I compute the distance between sample 19, say, and this cluster containing 2 samples? That's where this linkage criteria comes in. There's different ways to deal with that. So if I have a cluster containing multiple samples, and I have another cluster containing 1 or more samples, how do I apply my distance measure to get a single distance between those two clusters? We're going to use 3. There's the average linkage, which says if I have a cluster of m features and a cluster of n features, compute all the pairwise differences between those two clusters. So in the case here, I would compute the distance between 19 and 2 and the distance between 19 and 9, and I would average those distances. That would be my single number for quantifying the distance between sample 19 and the cluster containing samples 2 and 9. Average linkage. Complete linkage says I want really strong evidence of similarity between two things before I join them together. And so it uses the maximum of all those pairwise differences. You're not going to be called similar or close unless you're close on all comparisons. So I need the maximum to be small before I join things. And single linkage says I will call you similar if you're similar in any respect. So take the minimum of all those differences, or distances. These are the 3 most commonly used linkage criteria: average, single, and complete. The other main or widely used algorithm for cluster analysis is called, well, there's several, but the other that we're going to play with is called k-means clustering. You can do Bayesian clustering, you can do all kinds of algorithms. K-means is widely used. K-means operates differently from hierarchical clustering. Basically what it does is, given like these 2-dimensional data, we have m-dimensional data, where m is really big. If I just had 2-dimensional data, x coordinate, y coordinate, number, I might be interested in grouping those pairs, coordinates, together. And the way k-means works is if you tell me what k is, that's one of the limitations. You have to tell me what k is, what k to consider. So if you tell me that I think k, the number of clusters, is 3, then the algorithm will go and try to join the samples or the features together into the optimal 3 categories. Roughly speaking, the way that works is some centroids are generated, so blobs that I pick throughout the picture that I then go and group around. So if this is one centroid, I'm going to group these individuals into that guy's category if they're closer to that centroid than they are to the nearby centroids. So initially, I wind up with a clustering like this, red, blue, green. I then iterate by changing the centroid to be the centroid of each of those clusters and rejoin again, or recluster again. I repeat that until convergence, until they're not moving anymore. And I wind up with a picture like this. It can be for greater than 2 dimensions, of course. It's easier to see with 2 dimensions. But generally, with m dimensions, I wind up with k centroids, and each of my individuals has been grouped into one of those k classes. So one of those 2 algorithms, we're going to use both of them. We'll focus mostly on hierarchical clustering, but both of them are interested in the same thing. Let's group features or samples together into categories within which they are similar according to some distance measure. All right, we're out of time. What we're going to do next time is play with this script. It's called cluster example. And for a toy example, came from the Wikipedia article we were just looking at, let's walk through how clustering works. So first we're going to compute all those pairwise distances between our features, and we're going to choose who goes together first, so who gets joined first based on who had the smallest pairwise distance. And then we're going to work through iteratively, bit by bit. Once I've got that initial pair, what are the distances between all the clusters where one of the clusters has a pair, everybody else is a single individual. Join again, build another distance matrix that has now 2 clusters and individuals, join again, join again, join again, until there are every feature joined together. And then we can make a picture like we did a moment ago for the yeast data, where I plot a dendogram and interpret it. So after we do this example, you'll be set to do your homework, because I ask you to do some manual calculations in the homework for doing cluster analysis. Okay, so I will see you on Friday.